right now. So we are recording, and thank you very much, Heath, for agreeing to give a Build a Cell seminar, and it's all yours.
Well, thank you, Kate, for having me give the Build a Cell seminar today. I do really appreciate it, and I've enjoyed the series over these many COVID months, and getting to hear everybody's talks have been impressive. And so hopefully I can at least keep up to the tradition of what everybody has set forward with this seminar series. So for those of you that don't know me, my name is Dr. Heath Mills. I am the Chief Scientific Officer for Rhodium Scientific. My background is through academia. Got my PhD from Georgia Tech in applied microbiology, where I focus most of my work on extreme environments, looking at different extreme environments on Earth, which then led me into positions suited for space with a new extreme environment. And in Rhodium Scientific, I am guiding the company right now through with new markets, through the biotech sector, trying to get new initiatives built to better commercialize the low Earth orbit microgravity environment. And with that position, I'm also the lead for NASA mission integration and operation to make sure that the flight projects that we design and development are feasible for flight and then operate them while they're up on station. And so that's my role here with Rhodium, and that's who I am. And glad to meet everybody. My talk today is titled Synbio in Space. Yes, we are going to space for this talk and looking at ways to develop and further refine and move Synbio into the next frontier. And I don't even apologize for the space puns. When you're giving a space talk, man, you got to come up with a bunch of space puns all the way through it. So if you want to do a clicker and count your way through, it could be interesting. So with that intro and title, let's jump into it. And what is fun and what's happening now around us, and a lot of the hype from this summer's launch of SpaceX, with the Bob and Doug flying to the station, is that there is a new space race going on, and it's the commercial space race. And what that's allowing for research and for science and development sides is that the sky is no longer the limit. We can truly go to space. We can truly operate in space. But this time our mission is not just to go there. It's not just to go to orbit, but there are initiatives to go further, to go back to the moon, to put the next man and the first woman on the moon and go to Mars. And so in doing this, there's a lot of logistical challenges. There's a lot of things that have to happen to be able to make that leap possible. But from a humanitarian, from the humanity side of this is not the first time that we've made big leaps. We've taken these leaps before. Humanity made a big leap back in the age of discovery, where we left from Europe and left from foreign lands, and we landed here in the Americas. And we did this leap. And back then, the distance to the New World was as far as the distance that we're seeing now between here and Mars. And to make such a leap possible, one thing that humanity learned was that you had to use the environment that you were in. You could not bring everything with you. You could not pack everything up there and understand everything that was going to happen. You had to adapt. And so when the explorers came to the New World, they learned to use the land. They farmed the land. They raised animals here, grew their food here. They even used the land here for shelter and built their environment out of the local resources that they found. And they even healed their wounds and treated the sick from environment and from the resources that they found in this new world. Well, in making this new leap forward, the other thing that the colonists and the explorers realized was that not only did you have to go there and survive and thrive in that new world, but it also kind of helped if you understood that if you brought resources back, and if there was a promise to bring resources back, finding that funding, finding that means to get to where you want to go became a little bit more easy. And so in doing that, when we make this next great leap forward into this new environment, we have to think about these things. How do we use the environment that we're in? And then how do we deal with the limited resources that are available to us? Because when we go into space, there's no room for farming. And I put a little asterisk there because technically we may do some farming on the way, but it won't be the traditional farming that you see, and especially sitting here in the great state of Texas, farms are a little bigger than normal. But you can't do that in space. And so we have to think and retool our ideas as to how we can live in this new environment. And especially for this group today and who's listening here today, maybe Synbio is the answer. Maybe there are ways that all of these squares I have on the left, and then finding the food, finding the resources, finding those medicines, may be answerable in space by applying what we have been talking about over this whole seminar series, now just putting that into the space environment. And so check off the next space pun here. Here it goes. So going where no Synbio has gone before, and even threw in the little enterprise there on the left side for you. But the question then becomes why? Why take Synbio? What can we do with Synbio in this environment? And I really want this talk today to be less of a traditional research talk of, hey, here's what we've done, here's our stuff, our paper is now being published and wherever. I want this talk today to be more of an inspirational and a cross between a locker room talk to an inspirational review of what's out there. And so with that, I'm going to address three main areas of where we see and where Synbio may play a role. And the first one of those is through manufacturing. And this is something that may be more readily obvious to everybody, is using Synbio to produce a given product. The next one is using Synbio as an effector and using Synbio as a mechanism where a molecule is produced, a signaling molecule is sent out from the Synbio system that now changes the behavior of a natural cell, whether it's a bacterial cell, a prokaryotic cell, eukaryotic cell, a human cell. It provides a means for that effector to change the behavior of that natural cell system. And then the last one, and this is where it brings back to, let's bring something back to those here on Earth, maybe using the space environment for bioprospecting of novel genes, operons, structures, systems that now can be reintegrated or integrated back into a Synbio system that may change something here on Earth that goes into an industrial side or a medical side. And so those are the three main areas that I want to talk about today. So let's jump right in. Let's do the manufacturing first. And as I said, this is something that maybe is more on the forefront of your mind, is that you've seen these pictures. We go to Mars. We have either robots running around for us or a Synbio R2D2 style where we add in a genetic element, the organic machineries, the enzymes on demand to produce a given product. Maybe it's a nutraceutical, maybe it's a nutrient supplement, maybe it's a medicine, maybe it's a plastic, maybe it's a biofuel. But using this to really increase the resource acquisition in this environment. Use those local resources that you have, the CO2, maybe the silica, maybe whatever, but using that to produce something from a Synbio platform. Taking that one step further, you can even envision using Synbio to disrupt the need for supply chains. Rather than sending raw materials to the new environment, you beam the data necessary to print either a DNA sequence or a protein, or give it a new tool to that Synbio system by sending that data necessary to, hey, I need to print this vitamin. I need to print this new plastic. Can you please send me the data, the sequence, the DNA sequence, to be able to integrate that into my Synbio system? And now I'm able to produce that in a remote environment. This not only can work in space, but it can also work in remote environments around the planet. And some of you have talked about using Synbio in extreme environments around the planet where these supply chain issues may be an issue. So these are things that we can think about moving forward into space. Now, through the talks this summer, I've enjoyed hearing what everybody is making, what everybody is able to use these build-a-cell technologies to produce these new materials that may help with exploration, new molecular engineering, new therapeutics. Well, in space, there's still some work to be done. Some of these models that you're developing, it would be interesting to go fly these. to see if these systems are properly hardened to deal with microgravity and the radiation that's experienced on these environments. How does the new physical structure of that environment, with the lack of convection, the lack of flow regimes, the change in no sedimentation because there's no gravity, how do these systems need to be hardened for the microgravity space environment? Test the functionality of synthetic minimal cells, of cell-free systems. How do they work and operate in space? Can we tune those into this new environment? And then understand, can we introduce new genetic elements into these systems as we project that we may be able to? Let's demonstrate it. Let's show that these cells can be programmable on orbit to really show that reduction of this supply chain is possible. Now, again, I said I wanted this to be a little bit of an inspirational side and maybe spark some ideas. And so through this, and it's intentional that my email address is on the bottom down here, because hopefully as ideas start coming forward and are developed in your mind, and you can start saying that, hey, my project may fit what you're looking for. And obviously there's a lot more ideas than what I'm throwing on here. I'd love to hear these ideas, and I'd love to hear some feedback on this and start this dialogue, because that's how we get new ideas into space. And so please, through this, if you want to jot something down, send me an email. This is how I'd like for this talk to go forward. So that's the manufacturing side. It's pretty much simple, straightforward. Let's go to space. Let's show that this can work. Let's show the feasibility and functionality. Can we use some of the projects and some of the ideas that you guys have been talking about all summer in the space environment? But now let's take it a step further. We've gotten the Synbio systems working. Now, can we induce a new behavior with the natural cells? And so this is what we start off with, is that the Synbio systems are being ground tested. And we call it ground tested because now we're going to go up. We're going into space. There have been, and there are many systems, and we're actually working with Kate right now on a project where she's showing that using the Synbio as an effector on a natural cell works, and it works on demand and works with specific external cues. So we're seeing that this works. These can be tested here on Earth, but when we think about this system between the Synbio and the natural cell, we have two sides of this equation. We have the Synbio giving off a signal and the natural cell receiving the signal. Well, on Earth, we're setting this up as the Synbio is what we're building and the natural cells are what we know. Well, welcome to a new world, because the natural cells in space now are going to behave differently. This is a new environment for them. Space affects biology. And it does that by, and what we've seen over about 30 years of working in space and doing some good research in space, is that metabolic rates in space of the natural cells, so whether they're prokaryotic, eukaryotic, change. Some get faster, the metabolic rates increase, some rates decrease, but the biology changes. On station, pathogens, now some become more pathogenic, some less pathogenic. So the biology on that side changes, but also from a human research program standpoint, dosing to cure the illness changes. There are many studies that show the dosing of the medication needed on orbit is different than what we have down here. So maybe your Synbio system, natural cell system, is providing a dose-dependent drug delivery system. Well, in space, that now has to be re-examined because the natural cell is behaving differently. Growth in space is being hacked, and some cells that may be kept in check in a community environment, a pathogen that remains below detection limit is no longer a threat, now becomes a threat. So you may be dealing with, and you may have to now affect a different cell or different population than what you would be naturally targeting here. So now we have to go up because this environmental stress of space is creating a biological response that is not truly reproducible here on Earth. So we've got to go up, because now what used to be the unknown, which was a Synbio system, and now we are developing that and understanding a lot more, in this equation starts to become the known system. Now, the rules are being rewritten on the natural cell side because the natural cell responds to its environment. Well, this is a new environment. It adapts to this environment. Is that adaptation random, or can we predict it? The only way to get to predicting is to understand and have enough data to begin with. We've seen before where genetically modified organisms put into an environment to do a bioremediation study, all of a sudden that natural cell decides, I'm gonna go eat something else and do something else. And that predictable function that you had in the lab now becomes unpredictable in the environment. Well, welcome to a new environment. So now we have to, if we're gonna do an effector system, a symbio affecting a natural cell, we need to go up and start doing some testing, some real testing on this to understand how these systems work. So whereas the Synbio can be refined in space, and that's what we were talking about with the manufacturing side, it could be developed here on Earth. The natural cells, we need to go up and test those. And we need to go and do those studies to go forward. And so that brings me back to a quick slide again on what we need to go to launch and some ideas and some ways of maybe stimulating some conversation is that, and I mentioned the drug delivery systems, that efficacy needs to be rechecked. Does the same amount of synthetic minimal cells produce the same response on the same number of target cells, human skin cells, epithelial cells, take your pick cells? That needs to be reevaluated. A countermeasure study, let's say you have something that's dose dependent for radiation countermeasures or nutrient deficiency or a pathogen response. These countermeasures need to be revetted in this new environment. It's the same thing as going into the desert versus going into the jungle. Now you're going into space. Make sure that it's environmentally relevant. And then even down to the chemical reporter systems. Now you're talking about an environment that no longer has convective mixing. Convection doesn't work in space. Convection works on Earth because of density differentials. Heat rises, cold sinks, you got it, you understand basic physics 101. But now in space, heat stays where it is, cold stays where it is, you don't have convective mixing. So a chemical reporter system, is it as efficient to get the chemical from point A to point B? So things to think about. These are things that need to be addressed so that we can write the seminal papers that now then get reproduced and referenced for the next 20 years and everybody gets to stand on top of your shoulders. So that's that side. Now let's go next to the last one. Bioprospecting is fun. Bioprospecting now, let's use this environment to bring something back. Funding agencies love this one. Bring it back to Earth and save everybody here. And here's a little plug for myself on here. That's actually me over on the left side looking all engineering style, and as a genetics guy, I don't get to look like an engineer like that very often. But bioprospecting for me, especially in extreme environments, becomes really interesting and really, it's fun. And this was me out in literally the middle of the Atlantic Ocean on an integrated ocean drilling program vessel. We were drilling the mid-Atlantic ridge and collecting microbes from inside the Earth's basalt. We were looking for new life. We were going where no one had gone before, but this time down instead of up like I'm doing now. But it was out there, and it's from the Amazon, and it's from salt marshes, and it's from different environments that the whole field of bioprospecting has been going on for hundreds of years. And we know that we can develop new drugs, new nutraceuticals, new biofuels, new industrial applications from what has been produced and mined from biological systems. And we know that if we go into these extreme environments, we see novel biological systems. Well, on the left, that's inside the space station. That's a novel environment for biology. Bioprospecting on the station is completely untapped. It's not been done to the level that it can be done. And so what can we send up? Look at those adaptations, look at those changes. What can we then bring back down to see what novel molecular structures, cellular adaptations happen that we can now put back into a Synbio system? Maybe it's a secondary metabolite that's produced. A lot of our drug compounds, a lot of what we know as antibiotics came from stressing out biological cell systems, bacteria or fungi, stressing them out, observing what secondary metabolites they produce, capturing those, now turning those into drugs, and now we have antibiotics. Well, what can only be produced in this environment? We don't know. And I said before about identifying new populations. It may be a new pathogen that we haven't even considered yet, that right now on Earth is being kept in check by another member of the population, the member of the community, that in space, that community, that species that's keeping the pathogenic cell in check doesn't grow so well. Now it becomes an emerging pathogen. There's a lot of agencies right now looking for emerging pathogens. Space has an interesting tool on this, and I think that we can go and do something about it. So again, these are some ideas. I hope that you have 30, 40, 50 more. Email me, let's start a conversation on this, and let's see where we can take this. But so I talked a little bit about the why. Why go? Why go to space? What's unique about it? The manufacturing side, the using Synbio is exactly what we've been saying over the summer as an effector, trying to elicit a new novel and change the behavior of that natural cell. And then finding new ways of getting information from biological systems in a bioprospecting manner back into the Synbio system to now start developing new biotech tools. So let's go to space. It is actually getting to be that easy. And one thing that changes everybody's mindset, and I said before that I was environmental microbiology, I went in the field a lot. For me, it was an interesting realization that if you change the way you look at the space station as not just being the space station, but actually as a field lab, which is what it is, it now can become your field lab to do your research in this new environment. That changes the way you look at the space station. It is a lab. This is pictures inside the Destiny module, which is one of the US modules, one of the US tin cans that you see up there. Inside the Destiny module is the US National Lab. It has dedicated hardware, dedicated equipment, dedicated resources to conduct the research that you're right now thinking about in your head, that your lab is currently doing. This lab up here can do it now in space. So the facility is available. Now, and that's what we have, my fun part, man, left academia to jump into the commercial sector and to do some work like this, is that that's the fun part that I'm able to do here at Rhodium, is that we are a national lab, ISS national lab implementation partner. Through our commercial space services, we're able to fly these projects, not only our own, but yours. We partner with academic groups, commercial groups, government groups, to go and do research in space. Internal, meaning inside the space station, but also any unique platforms outside the space station, where now you talk about extreme environments, you talk about astrobiology, you talk about life in the extremes. Outside on the external side of the ISS, you can do your experiments. Times to launch are totally different thanks to the commercial side, thanks to Elon and Bezos and everybody and Northrop Grumman and providing rapid access to space and the new vehicles coming online from Sierra Nevada. We get timelines now where we can go from a kickoff of a project to launch in months, 4 to 6 months, even less if necessary. We can go. And so what we help to do, and what I'm having a lot of fun doing, is developing the projects, managing these projects from a space side, from the NASA perspective on this, the commercial side, doing that mission integration and operation. We even get into helping to do the grant writing, which as an academic, I thought I was going to get out of grant writing. No, still doing a lot of grant writing, which is, it works. And so we're able to help add text and add components to that. And the other fun part is that we've developed these Rhodium Science Chambers, which are an ISS science facility, which means, from a technical term, this is what you conduct your research in. They're on demand, they're customizable, and we can talk about details with that. But the main part in this is it's working and we're able to go. We've had a lot of fun this past year. Even through these COVID times, we've been able to launch 4 projects. That's me over on the left side looking all goofy. President and CEO and founder 𝗦𝗫𝗁𝗃 there in the middle, holding up what's about to go into space. What's going into space is actually inside that cooler, frozen, ready to go up. That's Dr. 𝗕𝗏𝗃𝗏𝗃 on the right side there. He's our program manager, and he helps develop a lot of our projects and work with the teams. Over on the right side, that was our employee for the day, astronaut Drew Morgan. working on station, working on one of our projects. We got to talk with him, visualize what he was doing up there, and help move his his hands remotely to make sure the projects were done on station the way that we had them designed. But our 4 projects that we flew, or 2 that we've flown so far this year, are phage evolution and space microbiome. And the last 2 flights are going to be in-flight biomanufacturing and space rhizosphere. And the other fun part is we get to go to space. We get to develop these cool patches, mission patches over here that are designed by the teams. But hopefully this has been a little bit inspiring as to what we can do and what can happen on orbit. And 𝗦𝗨𝗀𝗆's quote, I think, sums it up, that the journey of, and he said 1,000 miles, but I'm going to scratch through that and put 1,000,000 up there because now we're going to space. But a journey of 1,000,000 miles begins with 1 step. And what we're looking forward to doing is working with everybody to get Synbio to take that next step, to go into orbit, to go beyond, go to the moon, because this, I think, really, excuse me, can address, can address some of the challenges that are faced with going into space. And a real quick left side over here was the launch of NG-13, Northrop Grumman 13, back in February, launching one of the Rhodium projects into space. And getting to go see these launches are a lot of fun too. So I hope that this has inspired some questions and inspired some thought. And I do truly thank Kate for the opportunity to do this, for everybody giving us the chance to talk about this today. If you do have any questions, my email address has been plastered over this whole talk. And any questions right now, I'm happy to take them. And thank you guys very much for letting me do this.
Thank you so much, Heath. That was awesome. I have a few questions myself, but you've been getting questions in chat all through your talk, so maybe we can start with those.
Okay.
And if anyone has questions, please unmute yourself or ask in chat.
Let's see here.
You have, can you see chat?
I am trying to find it, Kate. I'll be honest with you. I'm trying to find it.
I think you need to exit full screen.
Okay. There we go. Okay. So, and yes, hi, Lynn. I'm glad you are here. And yes, there have been some Synbio. I had to stick to the Star Trek quote on that. You are exactly right. There has been some Synbio missions that have been going on, and I really look forward to talking with you about some of that data. So yes, so afford me that apology on that. I was trying to use a silly reference to Star Trek on that one, but yes, I will give the nod, and you are correct. There has been some Synbio work that's going in. I can't wait to see some of that data from that, and we will talk about that soon. Let's see, we will tell you about the power cell missions on the DLR satellite. Nice. So DLR is a great, it's the German Space Agency. Some other week. Okay, Lynn, we'll talk about that. Philippe, will antibiotic resistance not be an issue in a small population on spaceship? It sounds like it would be a huge danger. Did anyone ever study this? Antibiotic resistance is going to be an issue. It's an issue anywhere and everywhere. Understanding how antibiotics change and antibiotic resistance changes. There have been a couple studies on those, and actually I'm going to go back. I think it was a German study on that. But there have been some studies to show that antibiotic resistance changes. There may have been something out of Ames with antibiotic resistance as well. So yes, there is some data sets out there on that. It would be interesting to go in and address those studies further. Those synthetic cell experiments are a lot, are for space station, or can you do it with a balloon flight? There are a lot of good balloon studies and a lot of good programs using balloons to get up into microgravity or get up into lower, high altitude studies with higher radiation. I'm sure that there can be ways of doing synthetic cell experiments on balloon flights. I've worked on a project development for one of those before. So yeah, I'm sure that that could be a possible option there as well. Christopher asked, how does commercial space work? Boy, I need another 2 hours. Commercial space works in a very interesting way in that in developing a project, it is directly with a commercial company like Rhodium. Let me read the rest. How do companies make money by sending samples to space, or is it all for research so far? With being commercial space, there are fees, there are resources, there is cost to doing this. There are pathways to go faster. So commercial space works with matching up with an implementation partner like Rhodium to be able to develop the project. And then what we do is we pitch the project through the ISS National Lab, through NASA, and we get the allocation necessary to go to the ISS. It works through a fee basis on this, but this is something that we can talk about depending on what the project is and what the pathway would be. And there's different projects that go different pathways on that. Thanks for the amazing talk. I appreciate that comment. Great talk. Appreciate it. Where are we with respect to microgravity bioreactors? We are ready. There are bioreactors that are available, different sizes, different constraints, different requirements for the different bioreactors. There have been bioreactors that have been on station before. There were bioreactors that were developed all the way back to Skylab back in the 70s. There are, for different growth parameters, for mixing, fluid mixing, light requirements, CO2, take your pick with what you need to make the growth in the bioreactor work. There are possibilities for that. Email me, let's talk, and I can tell you the details on all of that. But there are bioreactors able to maintain growth on orbit for plant-based photosynthetic as well as eukaryotic, prokaryotic cell demands. Thanks, Ivan. Appreciate the comment there, and good seeing you. Good hearing from you. We should talk soon. Robert, good also hearing from you again, sir. Anna, thank you very much. What are limits for Synbio in space? Can you send multicellular organisms to? Limits in space. I always say that space is limitless, but there is limited space in space. But not so limited that you can't send multicellular organisms. Yes, multicellular organisms have flown before. Everything from flies, fruit flies, even up to rats. Rat team is a great project, and the rat research there, rodent research teams are really popular. Good work there. Down to nematodes and other multicellular organisms. There are some constraints, obviously, with size, with need of crew time on that, but love to talk about a multicellular experiment on this. It's definitely feasible. Even the biggest multicellular organism up there, they kind of like to have fun with us down here, and they're called astronauts. So they're even available at times to do some work and help us out. So let's see, Greg, what's the difference between satellite and ISS missions? Is this length? Biggest difference between satellite mission and an ISS mission is return capabilities. Right now with a satellite, there's not a return capability for your samples. So there's a lot of good companies out there that are developing CubeSats, satellite technologies to do even microfluidics, doing cellular experiments in space. Space Pharma comes to mind right now as a very good company that's developing some lab capabilities in space from a satellite perspective. But inside the ISS, if you move inside the ISS, now you have the option for return. And that's one of the biggest differences. Length of time in orbit on ISS, you can stay as long as they allow you the allocation with this. But also as a satellite, Depending on the height, depending on the size of the satellite, those can stay up for weeks, months, years, totally depends on the mission profile. So I guess the biggest difference on that is the return capability of your samples. If that's necessary, then I would suggest ISS internal to come back. Can you do a bioreactor manufacturing in space, like large bioproduction scale? What's the biggest bioreactor ever used in space? Great question. First part of that, can you do bioreactor manufacturing in space? The answer is yes. The size of this, typical bioreactors have been in the hundreds of milliliters up to liter size. They've not really gotten larger than a few liter in size, but that does not mean it is not possible, meaning there are more missions going up, there's more resources going up, there's more vehicles going up, and with the right use case on that, larger bioreactors, dedicated bioreactors can fly. We have some good insights in and good partners that we're working with right now that, like I said, for the right use case, then we can talk about larger scale bioreactors flying. And that's something that we are developing right now. So please email me. Let's talk about that one. That would be fun. Evangeline, thank you very much. I hope it was inspiring, and thank you so much, and hope to see an email about what inspired you on this one. Thank you much. Hi, Lynn. Good hearing from you again. So we've done some balloon missions over the years. Awesome. Thank you for adding and helping me on this one. 2.5 hours locally in the San Francisco Bay Area. Longer duration missions are possible towards the poles. Right. And the team that I worked with was actually doing something out of Antarctica. So thank you, Lynn, for helping out with the answer on that question. Isaac, are most of the missions so far focused on farming in space? No, farming was just an idea of talking about how do we survive in space. And so the farming side of that, you can think about farming and using, say, some of the bioreactors that we've been talking about to produce food stuff in space. But that's one side, the production of drug compounds, drug molecules. That side, even down to biofuels. There's been some biofuel work done, even down to production of plastics. We are actually flying a project coming up that will involve production of some plastics. So there's many different, a full myriad of different production capabilities on that side. So hopefully that answers your question. Can you send a student project to space? I read that first time. So you're saying, can you send a student to space? I had some of those I wish I could send. But a student project to space, absolutely. All the PIs are right now laughing at that one. They all have people in mind. But can you send a student project to space? Absolutely. And that's a college level student, but there's also programs to send K-12 STEM projects to space as well. It does not have to be a professional lab at all. This can be, like I said, it can be STEM, it can be undergraduate level, graduate level, and then it can be commercial government labs. Anybody, this is fair game for everybody. And Lynn, yeah, Lynn jumps in and says, no, not farming. Yes, true. Pam says, you said plant-based photosynthesis. Did anyone try cyanobacteria in space? Yes. So cyanobacteria within the bioreactors are absolutely and have been done before. There's been several bioreactors that's been maintained both from shuttle days up to ISS. And so photosynthetic microbes, bacteria are absolutely possible. Even say algae as well is very possible within these systems. So yes, and there are plant-based capabilities on station as well. But yes, cyanobacteria is a possibility. What about shrimp larvae? Can something be done in space? I don't see why not. There's been zebrafish that has flown before. I don't know if shrimp larvae has flown before, but I know that they have flown zebrafish before. And so shrimp larvae would fit obviously within that scale, that size scale. So yeah, Robert, I think that's definitely a possibility. Can you please name some places where such kind of research is going on? I want to join it as a PhD in space biology. The fun answer on that one is it can be done anywhere. Any research, any lab right now, with the right use case, with matching up the project that's done terrestrially, a lot of these can now be applied to being done in space. So you don't necessarily have to be at a single university or a single institute to do research in space. That is the fun part of the new commercial side of this, is that you can do the space-based research and you don't even have to be inside the US. I know that we have international partners here and people coming in from international areas. You don't even have to be inside the US. You can go through other agencies, European Space Agency, JAXA, Roscosmos, but then you can be in Europe and work with the US partner on this. We've flown European projects before. Lynn mentioned the German Space Agency. We've flown some German projects. So on this case, it's open to the ideas of your PI to get the projects to go to space. And then Lynn says we have done some cyanobacteria lysates because we need a very long multi-year stasis ISS. It's different. That's true. Absolutely. Yeah, so that's a good point, Lynn. Can you run HPLC on the space station? I have not seen, I do not know of any resource for HPLC on station. I'd have to look into that for you, Hans. I don't know the answer to that one, but I'm going to hesitate and say a soft no on that one. But that's something that we could look into and see. But soft no on that one. A lot of what happens with these studies and with these samples is that preservation of your sample on orbit is you then bring back the sample and you run the sample back in your home lab. And so you constrain the experiment to a growth sequence in microgravity, preserve the sample, bring it back down, and then run it back on Earth. So that may be a quick fix if there is no HPLC, but I don't think there is. And Lynn says Ames has had a series of satellite biology missions over the years based on microfluidics, including Genesis, Pharmacis, and 𝗕𝗏𝗄. Yes. And there's been a lot of satellite biology work. There's some projects that are going up right now. I know there's a launch coming up on September 2nd with a team from Italy and Israel that I've been following. There is many different projects that do satellites. Satellite is a good way of doing the biological missions. Having the right satellite CubeSat built with the right transmission requirements on that to beam your data back produces a great study and great results. I'm also interested in Mish's question about working in space biology. Okay, to that end, what kinds of companies, partners are involved in research in the space station? Is it mainly academic research settings or independent companies, or a mix of both? It's a mix of both, and thankfully it is a mix of many different entities, from government agencies to academic to commercial companies. Working in space biology and doing space biology is now being done by a wider group. A lot of how you pick which company or which academic group or which lab really depends on what questions you're asking and finding the right experts and the right pieces of hardware. If you have some ideas on that, if you want to ping me with an email, I can help maybe direct you to those that have the best resources on those. And we might be the answer to that, or might be able to find one of our partners that can help you out with that. Hi, Robert. We sent a seed sample, seeds from a school in Ecuador to the ISS. Okay, awesome. And see, this shows another international partnership there. Robert's doing a lot of good things down in Ecuador. I've had a lot of good chances to talk with him on his work. Their group is very active, and so there's some seed samples that they set up, the radish seed samples for a school. Good, Yvonne, thank you very much for adding the STEM set there, the link there for the STEM for students. Thank you for adding that link. Fly shrimp larvae, do experiments, and then have deep fried shrimp. The astronauts, I'm sure, would appreciate that. Living on the Gulf Coast, I wouldn't blame you. Really amazing talk. Thank you very much for that. What is rate limiting for ISS experiments? Takeoff weight or time on instruments on station? Harris, that's a really interesting question right there. rate limiting on this, I think on this would be, it's not so much size is limiting. So take off weight. I'm going to go with volume on that as opposed to weight. Usually it's a volume issue that we take up too much space on the vehicle going to space. I put space in the same sentence 3 times. But it's more of a volume issue on that. The complexity of the project, the number of manipulations that need to be done to complete the experiment on station can be a limiting factor. And what that boils down to is the amount of crew time that's required. Typically, if you can reduce the amount of crew time, then your flight becomes more feasible and operationally feasible for getting the right allocation to go to station. So making the experiment less hands-on, more automated helps out. And so there's a give and a take between the engineering complexity of that and the amount of crew time that would be necessary. That is a rate limiting factor to some extent. But again, and one thing that we try to make sure that we focus on when designing and helping develop the project is that we don't sacrifice the scientific output so that we fit the program or the capabilities on station. And so therefore we have we don't want to lose the science for sake of simplicity. And so we really take the time to work with you to develop the project to make sure that the objectives are met and still maintain flight readiness. So it's a mixture of questions there, a mixture of answers, but I hope I addressed it enough to get the point across on that. And thank you very much. I appreciate the last comment there. Any other comments? Anything else? This has been a lot of fun.
Thank you so much, Heath. That was really awesome. For some reason, everyone likes to type in chat. Nobody wants to unmute themselves and talk.
I don't mind as long as I can read it on here and keep up with it, and I do appreciate it. And it's always good to get help and answers on the chat too. So I appreciate those that were answering the questions and helping out on that. Thank you very much.
I like the deep fried idea.
I know it's about to be lunchtime here in Texas, so that's inspirational for me for lunch options. I appreciate it.
Do they actually cook on station, or it's all freeze-dried rations?
There is limited capabilities for that. I know that there were actually, and one of the experiments that happened over this past spring was they actually baked cookies on orbit for the first time. So I'm sure the smells of the station improved that day as the cookies were baking. So there is some capabilities there. Okay.
So if we have no more questions, let's go eat cookies. And thank you very much. Thank you very much, Heath. Thank you so much. Thanks everyone for questions and for the discussion.
Thanks. Appreciate it, everyone.
And see you all next week. Bye.
Bye, everybody. Thanks.